Harald Sitte from Medical University Vienna speaks about transport proteins, from structure and function to mechanism.
Great, thank you very much. First of all, to the organizers for inviting me to this. Really, it's an honor to be here. Also on behalf of the Exner medalists, I have to say. And what I'm going to tell you about is a little bit about transport proteins and kind of closing the circle now. Coming from Giulio Sopatifurga's talk and over these two very beautiful talks, which kind of broke up the theme, and now getting back to transport proteins and also, so these people that currently work for my team, collaboration partners, mentors, and this is the team, how it looked like last year. And so the main goal of my research group is to understand how neurotransmitter transporters actually work. And here's a picture of the synaptic cleft where these transporters are sitting. And as you can see here, these are 12 transmembrane domain spanning proteins that are sodium chloride coupled, and their major function is to retrieve neurotransmitter molecules that have previously been released into the synaptic cleft back into the releasing neuron. And this is done by these proteins, the neurotransmitter transporters, and they take up these neurotransmitter molecules and repackage them via specific other transporters, the vesicular monoamine transporters, back into these vesicles into the neuron, and to make them available for another round of release. So that this is a relatively simple cycle that is orchestrating the signal transmission in neurons from neuron 1 to neuron 2. Because upon release of these neurotransmitters into the synaptic cleft, and then they are binding to the postsynaptic receptors, signals are transmitted. And these receptors are not only postsynaptically located, but also presynaptically, where they also act in feeding back to this releasing machinery, and also telling the releasing neuron how much to release and when to stop. So this is a very complex machinery, and the most important thing is that these transporters are in the main focus of my research work, and we are trying to, or aim to, understand a little bit about how they work and what compounds are actually targeting those. And so they are the clinically relevant targets of drugs. They can be used for drug treatment, and here you have a couple of compound families that are important. For instance, antidepressants that alleviate the symptoms of depression, but also amphetamines and cathinones. Cathinones also for depression treatment, and amphetamines, for instance, for narcolepsy or attention deficit hyperactivity disorder. Antiepileptics are also part of these. Tiagabin is one of the drugs that is used. But they are also sold illicitly and used for recreational use, as you can see over here. So these are the things that you can buy on the streets. And it's also amphetamines and cathinones, but also cocaine that targets these transporters and binds to it and elicits recreational effects, let's put it this way. And they are very popular, and still cocaine is the number one in popularity, although it's trafficking from the place where it's being produced to Europe and US and elsewhere is not easy. So these are people in my work, in my lab that do the work, and we have strong collaboration partners, as you can see down here on the left. And so you saw the face already once in your slides, Giulio. And I told you in the title that I want to tell you a little bit about the structure-function-activity relationship. And so more than 20 years ago, there was the first emergence of a permease structure that Ron Kabek's lab published, and that was really breaking the barrier. So he was setting the stage for this novel crystal era. And so it was Lac-Y permease, so far from what we are studying and trying to understand. But soon after, 2 years after, Eric Rowe was publishing a bacterial homologue of our neurotransmitter transporters. It is the leucine transporter crystallized from Acrifex aeolicus, a thermophile. This was a bacterial homologue, so it had features that were very similar to what our neurotransmitter transporters also have. And we were able to learn a lot from this LeuT structure. And then the same group, 11 years later, came up with the first structure of a human serotonin transporter, where he was really setting the stage for the new era. And soon after, so he published several other structures, and there are many transporters now already available as structures, serotonin transporter, but not of this family, not the norepinephrine transporter and the dopamine transporter, and we are eagerly waiting for those. Although for dopamine transporter we have already a Drosophila structure, which is the closest we can get, and it was also published by Eric Rowe and his group, by the way. And I want to turn to another transporter family today, because when we look back very briefly into the synaptic cleft, then you can see that this is mostly governed by the sodium chloride coupled transporters here, but these are not the only transporters that transport these neurotransmitters such as dopamine, norepinephrine and serotonin. So there's also another family, it's called the organic cation transporter family, and these transporters are also ubiquitously expressed, and they have been shown to play roles in various diseases and have been associated with, for instance, depression, with metformin uptake and diabetes, with colorectal cancer and oxaliplatin uptake. And Hermann Köpse and Giuliano Ciarimboli were really important players in this field to move organic cation transporter understanding forward, as well as Heinz Böhnisch, who was closely associated to the SFB special research program in Vienna, where he was part of our advisory board. And together with Lynette Dawes from San Antonio, I was also studying quite a bit on these transporters. And what we found out is, just to characterize these transporters, that in comparison to the norepinephrine transporter, where you can see that it has high affinity and low capacity, these organic cation transporters, here it's the subtype 3, can transport much more compared to the norepinephrine transporter, but at a much lower affinity. So it's kind of really clearing the extracellular space from any overload of norepinephrine, while the norepinephrine transporter proper is rather kind of being interested in the lower range of norepinephrine extracellularly. And when you compare these two transporters, then they are affected by individual ligands. For instance, corticosterone is affecting mouse OCT3 at high affinity, and atomoxetine, for instance, is a ligand that binds to the norepinephrine transporter. Both can thus transport norepinephrine. And dicinium-22 is an inhibitor that inhibits OCT3, but also at higher concentration, also the norepinephrine transporter. Why is this interesting? Well, because I'm now coming to one study, just briefly, where the link is also not based on the transported ligand, like norepinephrine, but where we showed that we actually have the possibility to use ASP+, which is a fluorescent ligand, to monitor transport activity of OCT3 at a very high resolution, but also that this transporter is actually not affected by any of these psychostimulant drugs that I mentioned before. So OCT3 is not inhibited at all, while corticosterone inhibits the transport of these transporters completely. However, when we measured efflux of, for instance, of any substrate from cells, then we found that there is a very delicate but complex interplay between psychostimulants like amphetamine and OCT3 in that way that it facilitates the efflux via OCT3. So there is some sort of interplay between psychostimulants that do not directly affect from outside, but apparently from the inside of the cells. But we then studied in more detail how these substances, and these are various compounds that are centrally active, coming from a cathinone like bupropion, which is used as an antidepressant, d-amphetamine, cocaine, but also ecstasy, and also we included methylphenidate. And what we found out is in this kind of screening assay that, for instance, modafinil is not binding at all, which is also an atypical dopamine transporter inhibitor, but methylphenidate, which has a very high affinity to dopamine transporter, also inhibits one of the subtypes of organic cation transporter at high affinity. So these two guys were actually performing all these experiments. But then we moved forward, and what we wanted to study and to understand is why do some of these compounds interact with both transporter types or families and others do not. And then we embarked on a study which was actually yielding much bigger results than we anticipated before, because first I met with these two former collaborators from Copenhagen University, Ulrich Geter and Freya Herbock, and they told me about a large data set from the iPsych consortium on transporter mutants or genetic variants that they had determined from neuropsychiatric disorder in Denmark, in a time where data safety was not that big in Denmark. And nowadays I think this would all be barred and not possible. So they told me about these genetic variants and asked me, would you be interested in looking at those and determining what their impact was on OCT3, it was specifically on OCT3. And I said immediately, yes, that's a great idea. Let's look at this and let's do it. And then we embarked and started making all these cell lines with all the mutants. And then we had the idea, well, why shouldn't we actually team up with Vladimir Khokhov from ETH Zurich and Paul Scherrer Institute and Kanapa, his postdoc, and determine the structure of the transporter in combination with some of the compounds that we found very interesting. And we also did all the functional assays here and backed up everything with the computational group of Thomas Stockner in our institute and his two PhD students, Ralf and Erika, and to finalize all the whole story. And what we came up with is the first Cryo-EM structure of a member of this family in its apo conformation, and you can see here that it's embedded in some lipids, and so we have a large extracellular domain which most likely helped us or facilitated the Cryo-EM structure formation or the structure determination, because it stabilized the conformation of the transporter so well that we did not need to interfere with the structure at all. We did not have to remove anything. We did not have to mutate any domains or anything. We just embedded in lipid nanodomains and were able to perform the Cryo-EM structure determination. And so it's following the typical major facilitator domain fold, and as you can see over here, and here's a little bit nicer representation of the fold, and then we moved on, and I'm really cutting a longer story short. We moved on with determining the structure first from our experimental data, and then we saw that we were not able to completely resolve everything from the extracellular large domain. And so you can see here that there are some moving particles or moving ends, loose ends, so to say. And then we related to AlphaFold, which was named today already. And this changed structural biology quite considerably. And here we used it as an aid to complement the part that is in the outside part, and therefore that really helped us move forward. And we had this final structure then, or this as the final structure, and then confirmed that it was stable in the membrane over 1 millisecond, and both ways. But then we related to the AlphaFold hybrid structure because it was giving us the better outcome, also in terms of looking at structure or the structural integration of our ligands that we were interested in. And the prototypical inhibitors of OCTs were the first compounds that we were interested in to understand how they bind there. And these are just very simple pharmacological assays that show you that both Dicinium-22 and Corticosterone can concentration dependently inhibit uptake of a typical OCT3 transported substrate. So then we moved on and compared to the other three transporters, and what you see here is that corticosterone is actually only an inhibitor at OCT3, but not at OCT1 or 2, only at OCT2 at very high concentrations. And while Dicinium-22 is a more promiscuous ligand which binds to all three transporters in a very similar range. And then we resolved the structure of first the Dicinium-22 bound OCT3. So you can see it, you can almost see it in here, and then see it here much nicer, and the corticosterone. And we try to solve, based on these structures, why we have these specific binding themes or pictures with exactly these two different inhibitors because of their specific binding for corticosterone and the Decidium-22.
Richard?
Wo muss
Der
ich?
Computer, ich weiß es nicht. Der Computer, der Bildschirm dreht sich ab.
It's boring, so we turn down the...
It says any key.
Any key. So we have these two different inhibitors, and what we want to understand is why are they so different? Because to me it looked very similar how they bound. But then again, so we looked at the structures, and this overlay will not reveal a big difference. And what we saw is, yeah, they look quite the same. So it is not the overall way of the impact that these inhibitors induce in these OCT3 structures. It must be the specific way these inhibitors sit in the binding pockets, and this is exactly what we then found out when we looked closer to the Decidium-22, how it was actually lying in its binding pocket, and compared that to corticosterone. We found out that the corticosterone much more intimately interacted with very specific amino acid side chains, and that the Decidium-22 was kind of more wobbly bound in there, so to say. So in the end, we were not able to compare it to OCT1 and OCT2 structures because they have not been made available then. Now we have all structures, and now we also know why specifically OCT3 binds more intimately to corticosterone, and it is exactly the specific interaction with these amino acid side chains that makes it or renders it more prone to be specifically binding to these and not able to enter the binding path and pocket of the others. And we also found another interesting and challenging feature because we found that there is a side path which is a lipid bilayer axis pathway maybe, where possibly ligands may enter these transporters, because there's always the discussion about diffusibility of compounds and lateral access to ion channels or transporters, etc. And this could potentially be an interesting pathway for compounds that are lipophilic but close to the membrane and then sneak in to finally find their binding pocket. So this is currently something we are highly interested in. Haven't found a way to really challenge it at present, but this is part of our ongoing working program. So then, coming back to the initial idea that made me think about solving structures and looking at more detailed structure-activity relationships, that was the Danish cohort. And the iPsych data was really a huge data piece and I'm just cutting it short. So the patients that were incorporated in this iPsych cohort were suffering from one of the major psychiatric disorders, and that is attention deficit hyperactivity disorder, autism spectrum disorders, bipolar, depression, schizophrenia. And unfortunately, we could not get the power that enough power to say that one of our variants that we examined was predictive of any of those diseases or disorders. But what we were able to do is, of course, to then map these genetic variants and learn structurally from them, and to actually learn whether they render the transporter less functional or higher in functionality, or maybe do nothing, and are just there, which is also possible. And as also Giulio mentioned before, so have you also checked your OCT3 status?
Okay.
Good, but maybe it's not really important. We don't know yet. But it's just to point out that OCT3 is indeed related to those 5 major psychiatric disorders. And so what we did was then we did a huge panel of functional assays, uptake assays, uptake inhibition experiments. We looked at the cell surface expression of these transporters and the genetic variants. By confocal microscopy, we performed FRET microscopy and did in parallel also Western blotting to really look at the translocation pathway mutations in the end. And this is just one of these huge data arrays that we produced, where this is the control. And I'm not going into the detail, just to show you that there's a huge and vast array of different activity patterns that govern these genetic variants. We also looked at how they are inhibited by the 2 inhibitors that I already showed you, D22 and corticosterone, and we found profound differences in the way these molecules interacted with them. And in the, this is just one example where you can see that some of these transporters or the genetic variants are perfectly expressed at the cell surface, and others are also showing up in the inside. And these were the ones that we examined more closely. We tried to then help and rescue these transporters with the help of pharmacochaperones, and also in the end we came up with some solutions how these genetic variants actually helped us understanding the structure-function activity relationship of these transporters. And this is just a short summary. So what I showed you were the first Cryo-EM structures of OCT3 in 3 different states at, I didn't mention that, 3.7 angstrom. Resolution in APO, D22 and corticosterone bound conformations. We confirmed that these are pretty valid because the molecular dynamics simulations that we performed helped us to build these or to find the most stable conformations and confirm them. We have the individual properties and mapped them to the transporter molecules proper, and showed that they are distinctively binding to the transporter, and the genetic variants then in the end helped us in solving the structural basis. So in the end, so this was something that really connects structural activity and structural determination with bench and bedside. So this is also the mission of a medical institution such as the Medical University, and therefore thank you for your attention, and I'm happy to take questions.